There is a repair crew inside your body that only comes to work at night. And I don't mean that as a metaphor. I mean it as documented, peer-reviewed biology. While you sleep, specifically during the deep, slow-wave phases, three distinct populations of stem cells run their most intensive regenerative shift of the entire 24-hour day. Your hematopoietic stem cells are renewing your blood and immune system. Your mesenchymal stem cells are rebuilding the microscopic damage that accumulated in your muscles, cartilage, and connective tissue throughout the day. And your intestinal stem cells are replacing the entire lining of your gut, a surface that processes every nutrient you absorb and houses approximately 70% of your immune system, replacing it completely every 3 to 5 days. This is not optional maintenance. This is the biological process that determines how functional your body is tomorrow, next year, and a decade from now. Here is what most people have never been told. That repair crew either shows up or it doesn't, based on biochemical signals that are substantially determined by what you eat, and when you eat it in the 2 to 3 hours before bed. Not in some vague, general sense, through specific, documented molecular pathways connecting the timing and content of your evening food intake to the activation of specific stem cell populations. There is a direct line from what you consume tonight to whether your body's most powerful repair program runs fully or stalls. Most people believe that supporting stem cells requires expensive injections at specialty clinics. Most people believe that cellular aging is simply inevitable, something that accumulates quietly until it surfaces as disease, and there's very little you can do to slow it from within. I understand why people believe that. It's what most physicians have been trained to communicate. But after nearly 15 years in emergency medicine and preventive care, I've watched the science move far beyond that framing. The research on circadian biology, nighttime nutrition, and adult stem cell function is one of the fastest moving areas in cellular aging science today. What the last several years of peer-reviewed work have established is considerably more precise and considerably more actionable than what most people have ever heard. My name is Dr. Liana. I'm an emergency medicine physician. What completely shifted my perspective on aging wasn't a single study. It was a pattern I kept observing in patients that I couldn't explain with standard clinical reasoning. Patients of identical age, similar blood panels, similar lifestyles, and completely different trajectories. One aging with strength and resilience, the other declining steadily. When I began seriously examining the research on what happens inside cells during sleep and what governs the quality of that process, the pattern became impossible to dismiss. The people aging better were often, without realizing it, doing things in the evening that supported a repair process most of us are unknowingly suppressing. That is exactly what this video is about. In this video, I'm going to show you precisely how 3 separate stem cell populations operate during sleep, and why their effectiveness is directly tied to your evening behavior. I'll explain the specific molecular mechanism connecting what you eat before bed to stem cell activation and DNA repair enzyme function. I'll show you the research, real studies with real measurements, that makes this concrete rather than theoretical. I'll give you the exact foods with exact doses you can begin tonight, and I'll tell you when to expect results, what to look for, and the precise mistakes that silently cancel everything you're working to build. Quick request before we get into this. I regularly see messages from people who've watched several of my videos without realizing they weren't subscribed. Could you double check right now? It's free, takes 2 seconds, and it helps this channel reach more people who want science-based health information they can actually act on. Now let me give you the framework that makes everything else make sense. Because if you don't understand why nighttime nutrition connects to stem cell function in the first place, the foods I'm about to describe will seem arbitrary. Once you grasp the mechanism, they become completely logical. Picture a large, sophisticated factory running 2 entirely different shifts. During the day shift, the facility operates in full production mode, assembling, processing, outputting. The machinery runs hard and wear accumulates. But when the day crew clocks out and the night team moves in, the entire character of the operation transforms. Production quiets, and a specialized maintenance team, engineers, molecular repair technicians, infrastructure specialists, moves through every section of the facility, patching cracks, replacing worn components, correcting programming errors, clearing debris. By morning, if the night crew performed its job properly, the factory runs at full capacity again. That maintenance crew is your stem cell populations. And the scheduling system that coordinates when they deploy, where they go, and how effectively they work is your circadian clock, a molecular timing system governed by proteins called clock and BMAL1 that orchestrate biological activity across virtually every cell in your body. Here's the part that changes everything. The night crew doesn't arrive at full capacity automatically. It has to be provisioned. It needs specific molecular materials to function. And if those materials are absent, or if the signals calling the crew to work are too weak, the maintenance gets deferred, night after night, year after year. That deferred maintenance is what we experience as aging, not as an abstract process, but as measurable, cellular-level deterioration that compounds over time. And here is where the science becomes genuinely surprising. There is a single molecule sitting at the center of how effectively your stem cell repair crew gets activated each night. Most people know it only as the sleep hormone, but what it actually does inside your stem cells is an entirely different story. Melatonin, produced by your pineal gland in response to darkness, has direct receptors on mesenchymal stem cells throughout your body. When melatonin binds to those receptors, two things happen at the molecular level that matter enormously for anyone over 50. First, a study published in the International Journal of Molecular Medicine by Huo and colleagues worked with human mesenchymal stem cells and documented that melatonin binding promoted cell proliferation by inducing the expression of SOX2. SOX2 is what cell biologists call a stemness transcription factor. It is the gene that keeps stem cells being stem cells, preserving their capacity for self-renewal and their ability to differentiate into multiple tissue types when repair demands it. When SOX2 expression drops, stem cells mature prematurely into a single specialized type and permanently lose the regenerative versatility that makes them valuable. Melatonin keeps SOX2 expressed. It keeps your stem cells capable of remaining stem cells. Second, melatonin inhibited premature cellular senescence in mesenchymal stem cells through the activation of SIRT1. You may have encountered SIRT1 in the context of NAD+ and longevity research. It is one of the primary anti-aging sirtuin proteins. When melatonin activates SIRT1 in stem cells, it delays their entry into what scientists call cellular senescence. And this matters more than most people appreciate. Senescent cells don't simply stop working. They secrete what researchers have named the SASP, the senescence-associated secretory phenotype, a toxic mixture of inflammatory cytokines and enzymes that damages neighboring cells and can push nearby healthy cells into senescence themselves. Every mesenchymal stem cell that enters senescence prematurely doesn't just stop contributing to repair. It starts actively contaminating the cellular environment around it. Melatonin slows that process considerably. A 2025 systematic review published in PMC consolidated the broader picture. Circadian rhythms and melatonin regulate the proliferation and differentiation of stem cells in skin, intestine, the hematopoietic system, and the brain. This is not a single isolated finding. It is a converging body of evidence from multiple independent research groups. Now here is the problem that makes all of this especially urgent, a fact most people over 50 have never been told. Endogenous melatonin production falls dramatically after age 50. By age 65, most people produce between 60 and 75% less melatonin than they did at age 25. The primary cause is the progressive calcification of the pineal gland, a process that begins in adolescence and accelerates continuously with age. By the mid-60s, many people's pineal glands are producing a fraction of what they once did. Think carefully about what that means for the mechanism I just described. The signal that activates SOX2, the signal that keeps SIRT1 engaged in your stem cells, the signal that prevents your mesenchymal stem cells from entering toxic senescence. That signal is arriving each night at roughly 1/4 of the strength it carried when you were 25. The construction crew is at the site. The phone just isn't ringing with enough clarity. And most people have absolutely no idea this is happening. This is precisely why what you eat before bed is not a minor lifestyle detail. It is a direct dietary compensation pathway for a biological decline occurring silently in almost everyone over 50. Here is where it gets remarkable. There is a specific amino acid, tryptophan, that travels through the bloodstream, crosses the blood-brain barrier, and arrives at the pineal gland as raw material for melatonin production. During daylight hours, with the light-driven serotonin pathway active, tryptophan converts predominantly into serotonin. But when darkness falls, an enzyme in the pineal gland called aryl alkylamine N-acetyltransferase activates and redirects the pathway. Serotonin converts to N-acetylserotonin, which then converts to melatonin. Magnesium is a required cofactor for that final enzymatic step. Here is the timing implication most people miss entirely. Tryptophan consumed 2 to 3 hours before bed arrives at the pineal gland precisely when darkness is activating the conversion pathway. The same tryptophan consumed at noon flows predominantly towards serotonin, not melatonin. It is not a small efficiency difference. It is the difference between delivering raw material when the melatonin production line is open versus arriving when that line isn't running. Same food, same nutrient. Completely different biological outcome determined entirely by when you eat it. Now let me layer in the second mechanism because this one operates on an entirely different stem cell population through a completely different pathway, and it surprised even the researchers who uncovered it. A study published in the Journal of Experimental Medicine by Velarde and colleagues in 2022 followed 14 people through conditions of chronic sleep restriction. What they found was counterintuitive. Restricted sleep didn't simply reduce stem cell activity. It actually increased the division rate of hematopoietic stem cells. More division sounds beneficial at first. More renewal, more output. But what kind of division matters enormously? Rapid, disorganized stem cell division outside the correct signaling environment of deep sleep doesn't produce beneficial renewal. It accelerates clonal hematopoiesis, the dominance of one or a few stem cell clones at the expense of immune diversity. That state is a documented precursor to hematological disease and cardiovascular risk in older adults. The takeaway is critical. It's not simply how much you sleep, it's the biochemical quality of the environment during deep sleep that determines whether the stem cells activating overnight are performing organized, beneficial regeneration or uncoordinated, potentially harmful overdivision. Here's where the overnight fasting mechanism connects directly. Hematopoietic stem and progenitor cells are held in a dormant state in bone marrow, partly by insulin and IGF-1 signaling. When blood glucose and insulin are elevated, those inhibitory signals remain continuously active. The cells receive a constant biochemical message: resources are abundant, conditions are stable, stay dormant. A 12-hour overnight fast lowers insulin substantially, removes that inhibitory pressure, and sends an entirely different signal. Resources are constrained. Activate the self-renewal program. The most direct human evidence for this comes from research by Abdelhakim and colleagues at the Abu Dhabi Stem Cell Center, published in Stem Cells and Development in 2025. 10 volunteers observed Ramadan fasting, 30 days of daily fasting, with blood samples collected twice daily at multiple time points. Fasting measurably increased the absolute count of circulating hematopoietic stem and progenitor cells, carrying the markers identifying the most primitive hematopoietic stem cells, the ones with the greatest self-renewal capacity and the broadest regenerative potential. Fasting mobilized them from dormancy in bone marrow into active circulation. Not a pharmaceutical agent, not an injection, the absence of food. That result is further supported by landmark work from Walter Longo's laboratory at USC, published in Cell Stem Cell in 2014, which documented immune system regeneration through IGF-1 and pKa suppression during fasting cycles. The overnight fast replicates that suppression environment every single night. The third mechanism, one that almost never surfaces in mainstream health conversations, operates on your intestinal stem cells through circadian timing. Your intestinal stem cells are the most actively dividing stem cells in your entire adult body. The LGR5 positive stem cells in the crypts of your intestinal wall replace the entire gut lining every 3 to 5 days, an extraordinary renewal rate that depends on these cells keeping precise pace. Research consolidated in the 2025 PMC Systematic Review established that their cell cycle is directly regulated by the clock and BMAL1 circadian clock genes, controlling the checkpoint at which a cell commits to replicating its DNA in preparation for division. Intestinal stem cell renewal is not random, it is scheduled. It is organized around a biological rhythm that your behavior either reinforces or disrupts. When meals occur at irregular times, when eating happens late at night, when bright artificial light prevents the melatonin signal that anchors the circadian rhythm, the BMAL1 regulation of the intestinal stem cell cycle loses coherence. The long-term functional consequences are less efficient mucosal renewal, reduced gut barrier integrity, and more variable nutrient absorption. Now let me give you the complete protocol with exact doses, exact timing, and the reasoning behind every choice. The first food is a 28-gram handful of pumpkin seeds eaten 60 to 90 minutes before sleep. Pumpkin seeds are the highest yield tryptophan source in the whole food category by a meaningful margin, approximately 576 milligrams of tryptophan per 100 grams, meaning a single palm-sized handful delivers roughly 160 milligrams directly into the melatonin synthesis window. But what elevates pumpkin seeds above other tryptophan sources is what accompanies that tryptophan: magnesium and zinc, both direct enzymatic cofactors in the melatonin synthesis pathway. When you eat a handful of pumpkin seeds before bed, you are not merely delivering the substrate, you are delivering the substrate, the enzymatic cofactor for the conversion step, and the mineral the final reaction requires, all simultaneously. Plain or lightly toasted, a small bowl, nothing elaborate. The second option, and the most mechanistically layered choice on this entire list, is 30 milliliters of concentrated Montmorency tart cherry juice, diluted in approximately 150 milliliters of water, consumed in the evening. A study by Burkhart and colleagues published in the Journal of Agricultural and Food Chemistry documented that tart cherries don't merely provide tryptophan as a melatonin precursor. They contain directly bioavailable melatonin. 30 milliliters of concentrate delivers approximately 13 to 17 nanograms of melatonin that enters circulation during the critical sleep window. That is not a pharmacological dose, but it is a meaningful dietary contribution to circulating melatonin levels at precisely the time your pineal gland, if you're over 60, may be producing 60 to 75% less than it should. The anthocyanins in tart cherries add a third layer. Controlled clinical trials using objective polysomnography measurements showed that tart cherry consumption specifically increases time in deep, slow-wave sleep, not merely total sleep duration. Two interactions to flag clearly. Tart cherry juice contains significant natural sugar, so if you have elevated blood glucose or diabetes, dilute substantially and monitor your response. And tart cherries modulate the CYP3A4 enzyme system, which affects statin metabolism. If you take a statin, discuss this with your physician before adding tart cherry juice. The third food is 30 grams of raw walnuts, approximately 7 whole walnuts, eaten plain or mixed with a small amount of plain yogurt. Walnuts provide roughly 150 milligrams of tryptophan per serving, alongside alpha-linolenic acid, the plant-based omega-3 fatty acid. Alpha-linolenic acid contributes to the phospholipid composition of cell membranes in stem cells. Healthy, fluid cell membranes improve receptor function, including the melatonin receptors on mesenchymal stem cells that the entire SOX2 SIRT1 mechanism depends upon. Store walnuts in the refrigerator. If they smell sharp, bitter, or paint-like, they've oxidized and are no longer beneficial. The fourth food is a medium-ripe banana consumed 60 to 90 minutes before sleep. What makes this worth including alongside the others isn't the tryptophan alone, it's the vitamin B6 content. B6, specifically pyridoxal phosphate, is a direct cofactor in the enzymatic conversion of tryptophan to serotonin and from serotonin toward N-acetylserotonin on the path to melatonin. The riper the banana, the higher the B6 bioavailability. This is the lightest, most digestible option on the list, low volume, high biological utility. For people who find any substantial evening food intake disrupts their sleep quality, this is the most elegant entry point. The fifth element is not a food. It's a supplement, and it earns its place here through a specific mechanism. Magnesium glycinate at 200 to 400 milligrams taken 30 minutes before bed. Magnesium is a required cofactor for HIOMT, the enzyme that performs the final conversion from N-acetylserotonin to melatonin in the pineal gland. Without sufficient magnesium, the conversion is enzymatically limited at that final step. You've stocked the production line with raw materials and then removed the tool that completes the final assembly. Separately and independently, magnesium glycinate objectively improves deep, slow-wave sleep, documented across multiple controlled sleep quality trials. The glycinate chelate form has the best gastrointestinal tolerability, particularly relevant if other forms of magnesium have caused digestive discomfort in the past. A note on exogenous melatonin supplementation, since many people watching already use it. The receptor pharmacology research is counterintuitive here. Most supplements are sold in 3 to 10 milligram doses, but 0.5 to 1 milligram is both sufficient and substantially safer for consistent use. Higher doses over time can desensitize MT1 and MT2 melatonin receptors, producing tolerance and ultimately reducing your sensitivity to both supplemental and endogenously produced melatonin. If you use exogenous melatonin, 0.5 milligrams is the evidence-matched dose, and do not combine it with benzodiazepines. For warfarin users, the anticoagulant interaction is documented. Discuss with your physician before starting. If you take antidepressants in the SSRI or MAOI class, discuss any change in tryptophan intake with your prescribing physician before implementing this protocol. The sixth and most overlooked element of this entire protocol is the overnight fast itself. Finish eating before 8 p.m., ideally between 7 and 8, and consume nothing until breakfast. Water and unsweetened herbal tea don't interrupt the metabolic fasting signal. Everything else does. The nightly glass of wine, the piece of chocolate, a glass of juice, each re-elevates insulin within minutes, reinstating the inhibitory signal on your hematopoietic stem cells. The target is a minimum of 12 hours between your last food and your first meal the following morning. Consistency matters more than rigidity here. 3 to 4 nights per week of maintaining this window produces meaningful cumulative benefit, while 5 to 7 is the ultimate goal. The composition of dinner matters directly to this mechanism as well. Protein-rich meals stimulate glucagon, which has metabolic effects broadly opposing insulin and doesn't suppress hematopoietic stem cells in the same way. Refined carbohydrates at dinner, large portions of bread, white rice, pasta, or dessert, generate the insulin spike that directly undermines the overnight fast's ability to reduce stem cell inhibition. Protein and vegetables for dinner. If you need carbohydrates, keep them low glycemic. And consistent dinner timing, eating your last meal at the same time each evening within a 30 to 45-minute window, gives the clock BMAAL1 system the environmental anchor it needs to synchronize intestinal stem cell activity to the correct circadian phase. Here are the clinical insights from years of working with patients on recovery protocols that consistently separate meaningful results from average ones. Tryptophan timing is everything, and most people get this completely backwards. If you eat your tryptophan-rich food within 20 minutes of lying down, the compound hasn't had time to absorb, cross the blood-brain barrier, and arrive at the pineal gland before the conversion window opens. Aim for 60 to 90 minutes before sleep. That is the window where plasma tryptophan levels align with the enzymatic activation that converts it into melatonin rather than serotonin. Combining pumpkin seeds with tart cherry juice is not redundant. They work through different layers of the same mechanism, one delivering precursor, magnesium, and zinc, the other delivering directly bioavailable melatonin, additional precursor, and the anthocyanins that improve the deep sleep architecture where all repair activity unfolds. Together, they produce a more complete effect than either achieves independently. Alcohol is the single most counterproductive thing you can introduce to this protocol, and I want to be direct about why. Alcohol produces sedation, not sleep. Those are not the same thing. Even one standard drink within 3 hours of sleep reduces slow-wave sleep by approximately 25%, directly suppressing the growth hormone pulses that coordinate stem cell mobilization and blunting the deep phase where DNA repair enzyme activity peaks. If you are following this protocol while having a glass of wine immediately before bed, you are systematically undoing the biology you're attempting to support. These two habits are simply not compatible. Keep your sleep window consistent within about 45 minutes every night, including weekends. Your molecular clock becomes desynchronized with variable sleep timing, and when it's desynchronized, the windows for stem cell mobilization, autophagy, and DNA repair shift unpredictably and overlap less with your actual sleep period. Consistency doesn't require rigidity, it requires a pattern the biological clock can organize around. The most common mistake is eating a large, high-carbohydrate meal close to bedtime. This keeps insulin elevated during the critical early sleep window, directly suppressing both autophagy, the cellular cleanup process that must precede stem cell repair activity, and the growth hormone pulse that triggers hematopoietic stem cell mobilization. If you eat a heavy, starchy dinner late, you've already compromised two of the three mechanisms I described before you've taken a single bite of pumpkin seeds. The second mistake is expecting results within days. The studies I've described measured changes in circulating stem cell counts over 10 to 30 days of consistent fasting. DNA repair marker improvements take 8 to 12 weeks of consistent nutritional support. Structural tissue changes take 2 to 3 months because structural repair operates on a different biological timeline than blood chemistry. The people who abandon a protocol in week 2 because they don't feel dramatically different are making a timing error. The third mistake is taking supplements in place of whole foods. A tryptophan supplement delivers tryptophan. A handful of pumpkin seeds delivers tryptophan alongside magnesium, zinc, tocopherols, and a fiber matrix that shapes how every one of those compounds is absorbed, transported, and utilized at the cellular level. The whole food matrix is not merely a delivery vehicle for the active compound, it is part of the mechanism. Let me tell you what to watch for and when, so you know this is working. In the first 1 to 3 days, most people notice a change in sleep quality before anything else. Not necessarily longer sleep, but qualitatively different sleep, waking with a clarity and physical ease that wasn't there before. In weeks 1 to 2, morning stiffness typically begins reducing, one of the most sensitive functional indicators of systemic overnight inflammation. Between days 10 and 14, the HSPC mobilization research shows measurable changes in stem cell circulation with consistent nightly fasting. By month 1, high-sensitivity CRP typically begins trending downward, and in months 2 and 3, the cumulative tissue regeneration effects become apparent in the measures that matter most: wound healing speed, immune response quality, and recovery from physical and cognitive stress. Aging does not have to mean irreversible cellular breakdown. The patients I've watched age with real vitality, remaining sharp, physically capable, and immunologically resilient into their late 70s and 80s, are not genetically special. What they've done, usually without fully understanding why it worked, is consistently support the biological systems that were always designed to maintain them. Your mesenchymal stem cells have melatonin receptors right now, waiting for a signal strong enough to activate SOX2, engage SIRT1, and delay the premature senescence, accelerating silently in people who don't know this mechanism exists. Your hematopoietic stem cells are sitting in your bone marrow, held dormant by insulin signaling that a 12-hour fast would immediately begin to remove. Your intestinal stem cells are cycling every 3 to 5 days on a schedule that your dinner timing either supports or disrupts. These systems exist in you right now, every single one of them. They haven't stopped responding to signals, the signals just haven't been reaching them clearly enough. A 28-gram handful of pumpkin seeds, a consistent dinner time, 12 hours without eating, 200 milligrams of magnesium glycinate. The simplicity of this protocol is not a sign that nothing significant is happening inside. It is a reflection of how directly your evening choices connect to specific molecular mechanisms that research has now mapped at the cellular level.